Hello. So, actually, holding your breath will be important. So, once again, my name is Chris, and today I'll be presenting a project that I did as part of my master's thesis, supervised by Dr. Peter Wing, focusing on oxygen sensing and coronavirus translation and replication. And in the later bit of the presentation, I hopefully will answer why cellular context matters. So, while creating this presentation, I tried to make it as accessible to everyone as possible in terms of biology. So let's begin with defining some key definitions. Hypoxia is a drop in oxygen levels which all cells must respond to maintain homeostasis and survival. Upon drop in oxygen levels, human cells release hypoxia-inducible factors that change gene expression, allowing cell survival. Interestingly, the same factors can regulate viral infection, either promoting or inhibiting it. However, while some mechanisms are well defined, we still do not understand how many cases hypoxia regulates virus infection. So let's begin with a quick overview of the two main mechanisms how hypoxia might affect virus infection. If we have a RNA virus infection, the cell will detect the foreign RNA and lead to interferon production. For the sake of this presentation, just know that interferons interfere with virus replication. Thus, the more interferons we have, the less virus will be able to infect and establish infection. However, in some RNA virus infections, like Zika or dengue, hypoxia-inducible factors actually reduce RNA sensing, leading to reduction in interferon production and thus better virus replication and worse prognosis for the patient. Another mechanism how hypoxia could affect virus replication is in case of DNA viruses, where hypoxia-inducible factors will directly bind to the virus and promote its replication. Thus, we have two clearly defined mechanisms how hypoxia would promote viruses. And in terms of coronaviruses, which can cause severe illnesses, and probably you know SARS-CoV-2, how they can cause a mild illnesses, which all of us probably had because they cause common cold. These coronaviruses have RNA genome, so we would suspect that hypoxia would promote their replication as well less sensing. However, we see a completely different picture of hypoxia inhibiting coronavirus replication drastically. So fundamentally, we have a question of how and why this happens, and why this happens despite of pro-viral situation in the cell. So one of the first experiments that I did, and I will walk through it, so hopefully everyone will understand, is I looked into viral translation in hypoxia and normoxia in cells that express all of the genes. And what I was able to show that the cells were constantly down-regulating virus translation, a key virus life cycle stage. Then I had the cells which did not have RNA sensors needed for coronavirus detection. The process was relatively similar. And the same was in the cells that can't respond to interferon stimulation, indicating that hypoxia reduces coronavirus replication in mechanisms independent of innate immune system, as we understand it. So to better grasp this, we can look to the infection through coronavirus perspective. So if we imagine this as a cell and the path is the infection establishment, coronavirus sees this normal oxygen levels as a path of some interference like RNA sensing or, for example, interferons. However, as oxygen levels drop, the path becomes much more difficult and the infection establishes way slower. This is a fundamental change in cellular response independent of our immune system. And indeed, when I did some experiments to proof of concept, I was able to show that hypoxia downregulates possible two proviral factors and downregulates one antiviral factor. While it is a bit counterintuitive that it inhibits antiviral factor, it shows that this interaction is highly difficult and not very easily understandable with just one gene. Interestingly as well, which I'm not showing here, is that we see that these interactions are conserved across coronavirus family, which is an important feature, meaning that hypoxia has evolved with coronaviruses and their response is very interlinked. So briefly in conclusion, I was able to show that hypoxia inhibits coronaviruses independently of RNA sensing and interferon stimulation, and that this happens as the cellular changes. And hopefully that will explain why hypoxia-induced factors matter for cellular context, as hypoxia-induced factors can be activated not only by lack of oxygen, but by infection, cellular stress, immune response, or growth, meaning that these factors can be seen in multiple stages of a life, and as well, hypoxia-induced factors will regulate multiple stages of a life. Whereas when we test and measure in the lab cells, we have to take into consideration how the cells would be in the organ or the tissue, what would be the temperature, the oxygen, the water tension. So hopefully I explained why the cellular context matters. Thank you for attention. And just before, I was allowed to say a quick word. As a new LGBTQ officer, I also encourage everyone to reach out if you have any problems or want to speak up or have any ideas of what we could do as part of LGBT community. But especially, I also will have a small drop-in session in common room. I will bring some biscuits, so please do come along and let's just chat on what we can do here. So, thank you.